So related to broad spectrum activity, when we talk about biocidal activity, one of the things that we have to look for is that the disinfectant does not have any gaps in biocidal activity within that spectrum. In other words, there's many disinfectant actives that might be particularly effective against certain class of microorganisms, but not so much against another one. An example would be quaternary ammonium compounds. It's very well known that quats have a activity weakness for gram-negative bacteria. This is why you typically see them formulated in conjunction with other biocidal actives to cover up that gap in activity. The second thing is that there should be no evidence of resistance development to that particular biocide. Clearly, that's of importance because if you're subjecting an infectious agent to a continual low level of that biocide and the agent is able to develop resistance, eventually that agent will not be able to be destroyed by that particular biocide. The third thing is that the disinfectant agent should be effective in short contact times. The reason that's important is because that's a proxy indicator for the relative biocidal power of the disinfectant. Disinfectants that are effective in short contact times have a lot of reserve power, and therefore when more contact time is allowed, they will have a better ability to provide total microbial reduction. The third thing is that the biocide or the disinfectant can handle soil and other adverse conditions in the environment, in particular dilution with suboptimal water of high hardness, for example, or high metal content. So this is also a very important aspect when selecting the disinfectant. Aside from the biocidal activity and broad spectrum, a second consideration for disinfectants is their safety. The safety to humans, safety to animals, and safety to materials. And essentially that means that the disinfectant products that we're using should not contain any carcinogens, they should not contain any sensitizers, they should be non-irritant to skin, to eyes, and to the respiratory tract, and they should be safe in both the ready-to-use form and the concentrate form. The best way of determining whether the disinfectant scores well in any of these dimensions is to look at the safety data sheet. In the safety data sheet, one of the things that we can look at is the health hazards table. When you look at that, there's going to be an easy numerical score that goes typically from 1 to 4 or 1 to 5. And basically what we look at is how high is that score. The highest that number, the least hazardous the chemistry will be. So for example, if you have something that has acute toxicity of 1 or 2, that means that it is very hazardous, as opposed to something that would have acute toxicity of 4. The other thing that you can see from the safety data sheet is whether the disinfectant contains substances that are carcinogens, that are sensitizers to the respiratory tract or to skin, or that are reproductive toxogens, or produce any other type of health adverse effects. All of that can be seen in the safety data sheet. Finally, another important criteria for the evaluation of a disinfectant product is its effect on the environment. And basically here, we're talking about the disinfectant being at least readily biodegradable, that the disinfectant does not accumulate in the environment and on living species in the environment, that it does not affect pit or lagoon biology, and as we mentioned before, that there is no evidence of resistance development, because as we said, the disinfectant typically will end up in the environment somehow. So we don't want environmental species to develop resistance because of the presence of the disinfectant. So one important aspect of safety to the environment from the disinfectant is the effect of the disinfectant on the microbial flora of a wastewater treatment lagoon. There's an interesting study from Korea where they looked at an anaerobic lagoon producing biogas. And they subjected the bacteria in the lagoon to very small concentrations of quaternary ammonium compound in the order of 40 to 80 ppm. Remember that in disinfection, we're talking about active concentrations of quat of about 1,000, 1,200 ppm. What they found was that at 40, 80 ppm of quat, there was up to 70% inhibition of the acetogenic bacteria producing the biogas, meaning that the quat made its way to the lagoon, adversely affecting the flora processing the water and therefore producing the biogas in the lagoon. Another study that looks into the effects of the microbial population responsible for wastewater treatment comes from Bodek's group in Europe. They actually looked at a system where they had reduction of chemical oxygen demand by bacteria in a controlled laboratory environment. Now, you will recall that chemical oxygen demand is one of the key parameters that the Department of Natural Resources or the local EPA looks at as far as the quality of the wastewater that we're putting out. They found that 300 ppm of chlorine, and remember, in disinfection, we're talking usually about 1,000 to 2,500 ppm of chlorine, but 300 ppm of chlorine in that wastewater actually inhibited the ability of the bacteria to process the COD, or the chemical oxygen demand, by levels of up to 70 or 80%. To me, this is a very important finding. So in talking about the selection of disinfectants, we also talked about the importance of the application method. So in application, we're looking at disinfectants that are easy to apply, that are visible upon application so that you can avoid spots or avoid reapplication and therefore wastage of products, and that the product strength is easy to validate, especially when one is preparing it from a concentrate solution. For example, the use of strips that are color-coded and give you the color and concentration of material when immersed is an easy way of validating the concentration of disinfectant. Concerning foaming and cling time, foaming and cling time is not only a property of the disinfectant chemistry, but it's also important concerning the method of application. In other words, you could have a disinfectant chemistry that has high ability to foam and has high ability to cling to surfaces, but if one is not using the right application method, it might not result as one would expect.